Recording presentation. Third time's a charm. Hello, everybody. I am Maxime Eeraerts from Ento23 in Belgium, and I'll present to you the talk titled Pollination Deficits and Their Relationship with Bee Visitation or Cultivar Dependent in an Entomophilous Crop. So in an entomophilous crop, insects or animals are required to mediate pollination or to facilitate pollination. So this means that insects or animals transfer pollen from the anthers of a flower to the stigma of the same flower or another flower in order to facilitate the production of fruits and seeds or both. When it comes to this dependency of insect pollination in entomophilous crops, we know that this dependency varies greatly from crop to crop, and there are crops for which insects are not super important compared to other crops for which insects are essential in order to produce marketable yields. And for these crops for which insects are highly important or essential, we know that if there are no insects to visit the flowers of these crops, that we have like yield reductions of 80, 90 to even 100%. Recently, however, we also have been thinking or studying or trying to study or ask the question if this insect pollination dependency continuum is also applicable or valid for different cultivars of a single crop. However, this cultivar variability is very much understudied in open field conditions. There are some case studies that include more than one cultivar of a single crop in highbush blueberry, in faba bean, and in apple, but these case studies are rare. There also have been some crop-focused synthesis on crop pollination in apple and oilseed rape and faba bean. But here, if the authors want to look at the true cultivar effect, they often have to see that this cultivar variable is often confounded with study identity or study theme or study region. So it's difficult to separate these two. In addition, when there are these in these cultivar-focused studies, the link with observed bee visitation is often not included. When it comes to measuring the pollination contribution of insects, there are two important ways in which this is being done. The first one is by measuring the pollination service. So this is what the insects contribute in the field. And this is done by comparing an open pollination treatment, so the fruits and the seeds that originate that are produced by flowers that are open to insect visitation. And we compare these open pollination treatment with a pollinator exclusion treatment. So here the flowers are bagged and pollinators are excluded from visiting these flowers. And by comparing open and pollinator exclusion, we can then assess what the insects contribute pollination-wise in a certain field. Another way to assess the value of pollinating insects and to assess the yield deficits is by measuring the pollination deficit. And this is measuring how closely insect pollination matches the maximum pollination in a field. And here we add another pollination treatment, so the supplementary pollination treatment to the equation. And this is the supplementary pollination treatment is these are flowers that are open to insect visitation in a field, but we add pollen or we supplement pollen manually by hand to these flowers so that we can see what's the maximum or the optimum pollination of that field. And by comparing the supplementary pollination treatment with the open pollination treatment, we can then assess this pollination deficit. So that was the introduction, and I will now go over the questions, the main research questions of this study or this talk. So the first research question is, to what extent do pollination deficits occur in the two main high-bush blueberry cultivars, or two of the main high-bush blueberry cultivars? And the second question is, does bee visitation reduce these pollination deficits in these two cultivars? So in order to study this question, we conducted a systematic literature search, and we looked for all site-replicated observational field studies in high-bush blueberry in the cultivars Blue Crop and Duke. And we know from a previous review / meta-analysis that Blue Crop and Duke are the cultivars which are studied most commonly in blueberry. Another selection criteria for these studies is that they needed to include both an open and a hand pollination treatment in each field, and that they also needed to had to conduct a pollinator survey to see to assess the visitation of honeybees and wild bees in each field. And we then ended up with 11 unpublished and published suitable studies. From these suitable studies, we collected the raw data. So we collected the pollinator data, so both honeybee and wild bee visitation of each field, and also the pollination data, so fruit set, berry weight, and seed set of each field, both the hand and the open pollination treatment. And we requested all these data we requested these data as average values per field. And we then scaled all variables per study between 0 and 1 with the formula on the slide. So this is an overview of the data. We have 11 studies, which total 286 site replicates. And we have a study from Spain, a study from the Netherlands, and then a couple studies from Canada and multiple from the USA. So there's clearly a North American bias, which can be explained by the fact that that high-bush blueberry cultivation historically has started in Northern America. So for the first research question, whether there are pollination deficits across these different cultivars, we calculated the pollination deficit with the scaled variables, with the scaled values, as the difference between hand and open pollination. And if this difference is a positive value, this indicates that there's a pollination deficit or that a certain field is suboptimally pollinated. If this value is 0 or negative, it indicates that this field was well pollinated. We then tested a statistical model, a no-intercept model for each pollination metric with cultivar as a fixed variable to assess if the average pollination deficit was greater than 0 or different than 0 or not. So these are the results. We did not find a significant average pollination deficit for Duke for any of the pollination metrics. However, for Blue Crop, so the other cultivar, we did conclude that there was an average pollination deficit for berry weight and seed set. As you can see on the middle figure and on the figure on the right, in red, Blue Crop, the data of Blue Crop is indicated, and we see that there's an average that the average of this pollination deficit is greater than 0, which is not the case for Duke. So then on to the second research question, which is the relationship, if there's a relationship between pollination deficits and bee visitation. Here we again used the pollination deficit data of the first research question, and we then also scaled all pollinator data per study, and we constructed a total bee visitation variable, which is the sum of the scaled honeybee visitation data and the scaled wild bee visitation data. And we kept those two separate because we know that wild bees and honeybees have a different pollination efficiency, but we were still interested in the total bee visitation because we also know that honeybees are very much a useful pollinator in this system. And we then tested 3 different models per cultivar and per pollination metric, which was a null model, a linear model, and a negative exponential model. Within the linear model and the negative exponential model, total bee visitation as a fixed variable. And we then selected the best model with the AIC value. So for Duke, we conclude that the null model was the best model, so there was no clear relationship between total bee visitation and the pollination deficits. While for the cultivar blue crop, we did also conclude that for fruit set, the best explaining model was the null model. However, for berry weight and for seed set, we found that a negative exponential model with total bee visitation as a fixed variable was the model that best explained the variation between total bee visitation and pollination deficits for seed set and berry weight. And this negative exponential model is quite interesting because we can see that with increasing total bee visitation, the pollination deficits decrease more strongly compared to when total bee visitation increases even more, that this decrease in pollination deficits becomes less pronounced. So this brings us to the end of more or less the end of the talk. So for the first research question, we did find that pollination deficits are variable across cultivars, and this is in line with other case studies in blueberry and also in other crops. It's important to note that most commercial hybrid blueberry cultivars are self-compatible. So also the Duke and Blue crop from our study. So we can see that they have a relative high pollination success. For instance, for fruit set, on average, we did not detect that there's an average pollination deficit. However, we also found that for berry weight and seed set, there are differences between cultivars. And these differences, despite the fact that both cultivars are self-compatible, these differences can be explained by variable pollination demands or variable pollination abilities of different cultivars. So a certain cultivar might require more pollen to be deposited, or another cultivar might have less ovules or more ovules, or it might produce more pollen, etc. So we're still trying to explain these results. And then for the second research question, the relationship between pollination deficits and bee visitation, we conclude that pollination management in high-bush blueberry, and probably in other crops as well, should consider cultivar identity. And the importance of, or the interesting part about the negative exponential model being the best explained model is that it indicates that it's important to determine the optimal number of honeybees and wild bees to optimally pollinate a crop, or to manage crop pollination, to manage pollination of a certain crop. As you can see schematically on the figure, with increasing bee visitation, your pollination deficits, they decrease up to a certain point beyond which additional bee visitation does not further decrease these deficits. And it's important for sustainable and effective pollination management to try and determine this bee visitation threshold. So with this, I would like to thank all my collaborators and my funding agencies for their support. And I would also like to thank you for your attention. And yeah, if there are any questions, I would be happy to take them if there is time. Thank you.